Good afternoon, everyone. My name is Andreea Dumitrașcu, and I'm part of the Connect University team, and I will shortly present you the way today's Connect University session will run. Firstly, for those of you who are attending a Connect University event for the first time, this is the knowledge-sharing flagship initiative of DigiConnect, aiming to inform EU colleagues and the public about the latest digital trends. For today's session, we invite you to join us via Slido using the code neurotechnology. I repeat, the code for Slido is neurotechnology. You can comment and ask questions, and they will be allocated accordingly. Also, please use the hashtag ConnectUniversity when sharing insights on the event on social media. In case of any issues with Slido video or sound, we advise you to watch the session via the YouTube link and use Slido only for questions and comments. Without further ado, I would like to invite Mr. Viorel Peca, Head of Unit DigiConnect Future and Emerging Technologies, for the welcoming and introduction.
Good afternoon, dear guests, dear colleagues, dear DigiConnect and Commission colleagues. Please be welcome to this exciting Connect University session on restoring the intraneural simulation, the role in restoring sensorimotor. I'm really excited about this topic because I usually start my presentations with these iconic pictures from the Star Wars. I hope there are many Star Wars fans among you, where Luke is testing his new hand after his original one was cut by laser saber by Darth Vader. So for me, this picture is really capturing the essential characteristic after of the FET and now Pathfinder FET program. Really the science fiction that has become, thanks to the extraordinary researchers, and I would dare to our support as well, has become the science fiction that become the reality behind fiction, thanks to the technology and science. Today we have with us to tell us more about this really science fiction-like topic, we have Professor Silvestro Muchera. Professor Silvestro Muchera is acting both in Italy and Switzerland. So he's professor of biotechnology at the Scuola Superiore Santana in Italy, and as well in the École Polytechnique Fédérale de Lausanne, where he's holding the Bertardelli Foundation Chair in Translational Neuroengineering. Professor Michela, his alma mater is in University of Pisa, where he obtained his degree in electrical engineering in '96, followed by his PhD in, guess what, biomedical engineering from the same university in 2000. And then from 2000 and 2009, he was assistant professor of biorobotics and Scuola Superiore Santana. He had a brief stint in the US in 2007, where he was a visiting scientist at the famous MIT with a Fulbright scholarship, and from 2008 to 2011, head of the neuroprosthesis control group and adjunct professor of the Institute of Automation in Zurich. It's obviously Professor Dr. Muchera's interest include this development of neuroprosthesis based on the implantable neural interfaces. He's author of more than 300 Web of Science peer review, several international patents, and is associate editor of IEEE Transactions on Neural System. In other words, dear guest, dear colleague, we have today an outstanding guest. Professor Muchera, thank you very much for accepting our invitation today, and you have the floor. Professor, you are muted. Perhaps, colleagues, we can unmute Professor Muchera.
Okay. I guess now you can hear me. Sorry.
Loud and clear.
Very good. Thanks a lot. So, good afternoon. What I was saying is that I'm extremely happy to be here with you. And I'm very grateful, not only, quote unquote, only for the invitation, but also for the support that future and emerging technology gave me and to my research over the past 20 years. So the disclaimer, if you want, not only about the startup company we created, but also about the fact that FET was really and is extremely instrumental for our activities in neural engineering. So what I work on in my two labs is what we call neuroprosthetics or neurobionics, which simply means the possibility to bring basic science, in particular neuroscience, to clinical practice. And we do that for many different applications. Just to give you an idea, we work also with exoskeletons for stroke neurorehabilitation. But we also do many different things with different animal models. On the bottom, you can see a figure of a Nature paper we published on restoring locomotion in paralyzed people in the long term. And what we do, for example, here you can see also from time to time we like to play with other people on different topics. For example, we developed a very simple technique to allow people to control with trunk movements. You can see here flying drones. So we do a variety of things in the framework, if you want, of neural engineering. But today, what I'm going to show you more in details is how you can use electrodes implanted into the peripheral nervous system to restore different kinds of sensory, motor, and autonomic functions. We start exactly from what Mr. Pekka presented before for the restoration of grasping, moving then to legs, to lower extremity, to vision, and autonomic function restoration. So the idea is, first, we spent many years to identify a very good technology, the intraneural electrode that we are going to see in a few minutes, and now we are exploiting them clinically in different applications, from amputees to people with vision impairment, to people with spinal cord injury, etc. So let's start with the first example. So the idea is, as Mr. Pekka showed, we want to restore a very fine control of the human hand bidirectionally. And what is in particular missing right now is sensory feedback. So we are not able to provide sensory information to the amputees, and it is extremely important to do that. If I try to grasp a very fragile object, not a pen, think about an egg, I can break it easily if I don't have information about the force I'm producing. If I'm not able to light a match, if I don't have information about the position of my fingers, and these kind of things. So we've been working for approximately 15 years, try to identify the best interface with the peripheral nerve with a good trade-off between invasiveness, so how much damage you're generating. You don't want to do that much, right? Because if you want to be clinically applicable with your technology, and the selectivity. And the selectivity can be easily described with my ability to talk only with Mr. Pekka suddenly without anybody else listening. So it's a very one-to-one connection, intimate connection with the peripheral nerve. We've been working, I was saying, for 20 years, 15, going back and forth, up and down in this plot. There are many different options. We can talk later if you want in the question time. And then with Thomas Stiglitz, a colleague and a friend, professor in Germany, we identified this time electrode, and the idea is extremely simple. Again, simplicity can be the key for a good translation in clinical practice. So you can enter with a tungsten needle into the nerve. This you can see it as a nerve, leaving inside these yellow strips, which can be polyimide, in this case, a very flexible polymer, a thin one. And here you can see very small dots, the black ones, which are the active side, the electrical contact. So the idea is that when you have inside the the polyimide with the contact, you can be extremely selective and being able to reach small fibers. So we spent a bit of time validating this electrode in animal models with another colleague of mine, with Javier Navarro. And then with Paolo Rossini in Italy, we tested for the first time this electrode in a patient, in Dennis, a Danish patient who got the amputation approximately 10 years before the implantation. And here you can see a video with the procedure. You see the surgeon entering with the needle and penetrating the nerve. Okay, so I mean, for a surgeon, it's a reasonably simple technique. So again, simplicity is a key. Then the very first moment, it was we called the magic moment. So we switch on the stimulation and we ask the subject, do you feel something? There is something you are feeling at your phantom hand. Remember, the hand is no more there because of the amputation. And then it was able to refer using different black dots, different contact, different sensation at the different position of the finger. So that was really interesting, but for us, the goal was a clinical one. So what we wanted to see is whether it was possible for dentists to use and exploit this in a functional way. So the idea was to have dentists controlling a real prosthesis with sensors on the fingertip while grasping an object. The prosthesis was grasping the object, we were getting sensation of the fingertip, and then we were translating with a very simple amplitude modulation, a very primitive one, the information coming from the sensors into information current delivered in the nerve. And if you want, the idea is pretty simple. The stronger is the grasping, the higher is the volume. You're just pumping up and down the volume according to the sensation. And then we ask dentists to perform a huge variety of tasks. Here you can see on the left the staircase task. So we will ask him to modulate up and down the force he was producing, stabilizing the force at 3 different levels of force, so low force, medium, and high force. And what was very interesting was Dennis' ability to do that in real time. So here you can see with this red arrow, Dennis from time to time was making a mistake in the control, and he was realizing that he was doing too much force and was trying to stop and readjust it in real time. So he can exploit it. He can exploit it in real time. And we compared, you can see on the bottom left here, we compared it. Dennis is a unilateral amputee. So we compared what he was able to do with the natural hand, which is in this case the green plot, or with our prosthesis, with the sensory feedback tactile we were providing, the blue one, or only with visual feedback, the usual commercial prosthesis. And here you can see that we are reasonably close to the natural one. There is a problem here which is more related to the quality of the sensors, and I will tell you something in 1 second. So, but I mean, the message is you can exploit the sensation in real time and is reasonably similar in terms of functional results to the natural one. Then again, we started playing. Dennis was really a patient, in the sense that he was very kind and very collaborative. What I'm used to say is that they are the real pioneers because they donate part of their life for us, for science. And it was extremely pleasurable to work with Dennis. He was a very tough guy, very resistant. So we tested many different things. We asked Dennis to identify, for example, the shape of the object, whether it was a cylinder, a small sphere, like a very small one, or a baseball sphere. And he was able to do it with quite good performance. You can see on the bottom. He was able to recognize the stiffness of the object. So here you can see, for example, hard, medium, and soft objects. He was able to do it reasonably well, with also an improvement over time in the sessions. Here you can see Dennis performing the task. So we were asking him to grasp an object completely blind and acoustically isolated, and he was able to say hard, medium, or soft. Okay, so he was able to do it. And what was also extremely interesting was that we look at the way he was doing it. And if you look at that, you will see that it was not the final force produced is pretty much the same, What is different is the dynamics, the evolution. So Dennis was able to see, to understand more or less exactly as we do the evolution of the force at the different fingers and use this information for understanding the type of task that he was performing, the kind of object he was asked to recognize. Then we moved a bit more into the complexity. In particular, Calogero Oddo is a colleague of mine, an associate professor in Italy, and he developed this fingertip sensor, and we used the sensor during passive textural recognition. So there was a sliding of the fingertip onto different gratings, and Dennis and 2 healthy patients in micro neurography, so with needles acutely implanted, they were able to understand whether one or the other was more dense in terms of grating. And even more interesting, we asked 2 new patients you're going to see in 1 second to identify in an absolute way the type of material, completely blind again and acoustically isolated, they were sensing. So they were able, one of them even to have 6 different object texture recognition of real objects, like rubber, Velcro, paper, and this kind of things. So, it becomes clear that with a very simple technology, you can open up a huge number of possibilities. But then, I forgot to say, but this was an implant for safety issues was only 1 month. Okay, we wanted to see whether there was any issue, any problem. So the next step was clearly to see whether we could have more patients for more time, and this is what we did. We implanted 3 patients, Eduardo, Almerina, and Loretana, for up to 6 months between the end of 2015 and the end of 2017. And the first thing is that we did replicate all the results that we had with Dennis till the very end. So the electrode was still able to work properly. Even more, I can tell you that in the very last patient, Loretana, you can see here, the electrode were more than 80% was still working after 6 months, and they were very stable. So, I mean, the stimulation was able to provide the same sensation day by day to the patient, which is not that common. Sometimes with neuroprosthetics, you have to recalibrate, readapt more or less every day. What seems to us that this technology is pretty reliable and pretty robust. Not only that, the 3 patients we selected were very different among them. So, for example, Fernando, the one on top, had a huge phantom limb pain at the end. Almerina got, the second in the middle, got an amputation more than 30 years after the implantation, so it was a very old one. And Loretana got an amputation with an important damage at the medial nerve, one of the 2 nerves we were implanting. The interesting thing is that in all these patients, with some difference in the performance, everything was working. So, of course, when you start, you want to have the champion, right? Dennis was the perfect candidate for many reasons. But now, at least with these 3, it's still a very small number of patients, but we know that we can generalize probably the technology to different kinds of applications and patients. Here you can see Fernando performing one task in particular. That was a task designed by my colleague Marco Controzzi, and this is what he called the virtual egg test. So you are simply simulating with that box a very small egg, and the idea is that the subject is to move it from one place to another without breaking it, thanks to the sensory feedback. And the interesting thing was that even Fernando, who had a very, as I told you, very important pain, he was able to do it after some training. You can see here at the very bottom left, it is interesting to see that Fernando started the performance very badly because of this, because of the pain. And he was able to not to reach the performance of the other 2, but to become reasonably better and reasonably okay. So clearly, it's not going to work perfectly with everybody, but it can work to a very good extent for everybody. Then the next step for us, which could sound trivial, but it is very important, we moved for something which was not still implanted, this is what we're working right now, but portable. So we put all the electronics inside the backpack. In general, when you see this kind of presentation, there is always kind of behind the scene, there is a rack with all the electronics. So what we did for the 3 patients to put all the electronics inside the backpack, and the patients were going around Rome, driving, go to Ponte Milvio, the romantic locks for lovers, and so they were enjoying the city while using our technology. So it was really reassuring that we have enough robustness to do to the next step. And this is what we are doing right now. On one side, we are going for a large clinical trial, but what we are also looking at is the possibility to deliver more information. For example, position information is a very elusive one. We really need to have information about where our fingers are, but with our technology, it's very difficult. So what we tried to do was to have a combined solution. So the ones I showed you before with the tactile sensation, and then we took some of the electrodes which were providing not really useful information, and we trained very quickly the subjects, 2 subjects of the 3 I showed you before, to go for a remapping. So we were able to kind of teach them to use this channel for the information about opening and closing of the fingers, okay? And what was really interesting was that the subjects were immediately able to do it, to use it, to recognize very small or very large objects. Not only that, they were able to combine the 2 sensations, the natural touch sensation and the remapped position sensation, okay, to identify small or large, soft or hard objects. Again, this means that this technology can provide a lot of information, a lot more information that we can provide, we can testing right now. The interesting thing also was that we asked the subject to do that while doing another task. So they were kind of counting backward, these kind of things, what is called cognitive dual task, to be sure that they were not too focused on the task. That's something you cannot do in real life, right? You are not supposed to say thinking while you're doing something else. And there was no change in the combined solution, so they were able to master this even in a very complicated situation. Then the next step was to make even more complicated the way we are delivering the sensation. Okay, so as I told you, pumping up the volume is not really something we do physiologically. It can sound a bit unnatural to the patient. So we decided to team up with a great expert, Sliman Benzmaya, in neurophysiology of touch. He developed a very sophisticated model to translate mechanical force at the fingertip into electrical stimulation, frequency modulation of the sensation. So this was really a biomimetic sensory feedback for frequency, not amplitude, not the volume sensation. And then what we did was to combine, to test in one subject the frequency modulation, the biomimetic one, our amplitude, and the combination, okay? So both frequency and amplitude with Giacomo, a former student of mine, and Alberto, a colleague of mine in Italy. And to make the story short, I don't want to go too long, but the message is combining the frequency and the amplitude, you can achieve excellent performance, both in the virtual head task that I told you, making more natural the sensation. The subject will never feel the sensation really perfectly natural, but just to give you an idea, this is the volume, and this is what you can get with the hybrid solution. So it becomes more natural for the patient, which is in the long term very important because it makes less unpleasant to use this sensation. And also, they were able to have more embodiment. So they were able to feel this sensation and this hand as more part of their own body. So the message is you can do more in terms of sensation you can restore and the way you are restoring. You can do more sophisticated things, and this is what we're going to test in the next years. Another question which is always extremely common and important is, but you can do it also non-invasively. What is the real advantage to implant somebody? we have just one very preliminary test with only one subject, so it's really just a consideration for next steps, if you want. If you have the sensation done non-invasively or with our technology, in general, you can see here there is, what you say, off and off. There is no difference. You could even say that maybe the non-invasive is more effective. The difference happens when you have, again, a cognitive task done in parallel. Then there is no change in the invasive one, and there is a big change in the non-invasive one. Why? Maybe because the person is more able, it's easier for the person to integrate the sensation using the invasive solution to the non-invasive. Very preliminary, but that's something we're going to investigate with the next patients. Then, as I said, we moved from upper extremity, from the hand to the leg. And in particular, we are now working on making prostheses for transfemoral amputees, so prostheses with the artificial knee and ankle, so more or less all the leg. This is very important for this patient to have a very easy way to control it. And the solutions are still quite preliminary. So with Alberto and Federica in a framework of a project called MOTU, we are developing solutions for combining different technologies, in particular muscular signals, foot switch and inertial sensors embedded into the prosthesis. We developed a quite sophisticated control algorithms for identifying different states for the prosthesis. So ground walking, going upstairs, downstairs, different kind of slopes and these kind of things. So the message is you can really achieve very nice performance in the decoding. So what we are now going to do is to test this approach in real time for allowing the patient to have a more effective control of the prosthesis, for the leg prosthesis, together with something we did in the framework of another project funded by FET. We also created a startup company with my former student, now colleagues, Stanislav Popovic and Francesco Petrini. What we've been working on is the possibility of using the same idea of implanting time electrodes for the arm, for the hand, this time for the leg. And in particular, they implanted the electrodes into the sciatic nerve, making a closed loop from the sensors provided in a sensorized insole back up into the stimulation. So it's the same idea, the same protocol, but this time for the leg. First, again, the magic moment. You can see here the results they got in terms of sensation provided to the patients. It is remarkably good. They were also able to optimize the surgical procedure. You can see how many sensations in the feet, the foot they were able to provide. And then the next step was to see again functionally what happens. And it was extremely interesting to see that the patients were able to have more confidence in using the prosthesis, more embodiment, they were going faster, and they were reducing the metabolic consumption while using the prosthesis. Why? Simply because they were able to easily incorporate the sensation we're providing, adjusting their control in real time. So this means that, as I showed before for the end, in this case, even more, there is a clear clinical benefit for this kind of patients. So what the startup company we created is going to do is to exploit this. They got a European Innovation Council grant for this, to bring this to clinical practice in the next few years. But then what we decided to do is to move from sensory feedback, mainly touch and position in amputees, to other applications. The very first one, together with my colleague and friend Diego Getzi at the PFL, was vision restoration. So the idea was there are people working on retina prosthesis. There are also companies working on this. There are people working on cortical implants. There was a paper published a few months ago in Science, in monkeys on this. Why not implanting the electrodes into the peripheral, or the optic nerve, which is central actually, in the optic nerve. So we developed with Diego a new version of the electrode we called optic Celine. They have also not only the main shaft, you can see here, but also small wings to increase the number of fibers we can elicit. And then we made preliminary testing, and what was very interesting was that we were able to elicit cortically a lot of different sensations. So clearly, the selectivity that I showed you before for touch in amputees can also be achieved in patients, in long term, also in blind patients, for sure, right now in animal models. So for this reason, what we are now working on is our next step, which will be, we are now submitting the application for implants in patients, in one or two selected patients, again at Gemelli Hospital. The idea is to try to replicate more or less what we did for touch in amputees, also in blind people, using optic nerve stimulation. The next step, and we have a FET project right now going, is to go what is called, and I'm going to the conclusion, what we call electroceutical. So we have been working on peripheral nerve for sensory, we're working on the optic nerve for vision, but you can also use the autonomic nerve, which are the primitive nerves which are inside the body, connecting all the artificial organs among them and with the brain. Okay, if you think about the gut-brain axis, it's becoming very popular, is also due to this kind of autonomic nerves. So the idea of electroceutical is to implant the same kind of electrodes, but this time for the autonomic nerve, since we know that there are many things we can do. For example, we know that there is bad neuromodulation in the autonomic nerve for diabetes, hypertension, osteoporosis, even cancer. So this is clearly the next step for neural engineering in the next 15 or 20 years to try to move from sensorimotor also to autonomic nerve stimulation. And what we are working on, just one slide, in the framework of the New Heart project I'm coordinating with Vincenzo Leonetti, is to restore the vagus connection. The vagus nerve is a big branch, a big autonomic nerve connecting more or less everything to everything in the body, and is also related to the down regulation of the heart activity in healthy people. When you get a transplantation of the heart, the cardiac surgeon reconnect all the vascular part without connecting the neural part, okay? And this is a problem because clearly it's kind of, as Vincenzo told me once, driving a Ferrari without the brake, because the brake is the vagus nerve for the heart. You can be James Bond and you can do it, but in general, you get a bit of problem. So what we are trying to do is to create a neural interface for the regeneration and reconnection of the vagus nerve in order to reconnect in closed loop this vagus heart control, with the idea to provide a benefit in the long term. This will be done in animals in this project, but in the long term, we hope to be able to help transplanted patients to have a longer usability of the heart they got. At the same time, we are trying in the same project and with the same activities also with Fabio Recchi, another colleague of mine, to understand more how we can use the vagus nerve. You know, in general, the idea is often I put electrodes in and I do something, which could be limited. So what we are trying to do, for example, is whether you can extract physiological parameters, physiological information from the vagus nerve to achieve a closed loop control, so to stimulate only when it is necessary. This is what happens in general in healthy vagus nerves in people. So we made an experiment with Fabio. The idea was to record from the vagus nerve while providing different kind of physiological challenge. So altering blood pressure, respiratory challenge, so changing the way the animal is respirating, these kind of things. And what was very interesting was that we were able to have, in general, quite good performance in terms of reconstructing this information. So really, we can open up a new way of using this kind of vagus nerve stimulation in closed loop, again, mimicking what happens in healthy subjects. Finally, just 2 slides about another challenge we have right now. We have to use D-time electrodes not for sensory restoration, but for motor restoration in people who are paralyzed. It's the dual of an amputee. The amputee does not have the hand. A person after a spinal cord injury at the cervical level, at the neck level, has still the hand connected, but the hand is neurally disconnected from the brain. So there is no commands going down for the control. So what we are doing is to try to restore this brain peripheral connection. In particular, what we are working on right now is to use the same kind of electrode, the time electrodes implanted into the median and radial nerve of non-human primates, with the idea to see whether we can be selective in the stimulation. So the idea is, can we achieve different finger movements while stimulating? Can we restore, not only as we do in general with current technology, big grasping, like this one, I hope you can see it, but very different finger movement in a more selective and rich way. The preliminary results are very promising. I hope that Mr. Pekka is going to invite me maybe in a couple of years to show what is going to happen next. But also in this case, we are already discussing with a clinical center in Florence to see whether we can implant these electrodes also in patients. So let me conclude what I try to show you by convince you that stimulating the nerve, in particular intraneurally, penetrating the nerve, could restore a lot of function, could provide a significant benefit in the long term to patients. The first step is to increase in the future the number of the patients we are implanting for the bionic limbs, both for the hand and the lower extremities. And then to increase more, as I showed, the number of applications moving from animal models to patients, both for vision, for grasping restoration in paralyzed people, and for autonomic stimulation, in particular in transplanted people. And the idea is really we could change, as I said, with electroceutical, the field, providing benefit to a larger number of patients with different kinds of disorders, because as one of the pioneers in the field, Tom Mortimer, now emeritus at Case Western, told me once, when I was kind of a teenager, was a PhD student, if you can play with the nervous system, you have unlimited possibility. And if you want, this is really the beauty of neural engineering, you can have access to the nervous system and you can really try to help in many different ways. Let me just thank, I will not be able to read everything, but all these projects have a lot of collaborators at the PFL, at Scuola Santana, at Università Cattolica and Policlinico Gemelli, University of Fribourg, University of Freiburg, University of Washington, and ETH. So it's really a large consortium I'm coordinating or co-coordinating. And below you can see, of course, the different agencies and foundations are helping me. In particular, again, I want to thank, not for being flattering, but because I really mean FET. Another officer once said that FET can change your life, and in my case, FET did change my life. So thanks a lot, and of course now there is time for any question you may have. Thanks a lot.
Thank you. Thank you very much, Professor, for this really, really amazing presentation and insights. I think it's also a wonderful presentation, representing what we mean with foundational technology. So once you have developed the basis, then you started with the hand, then you went for the leg, then you have for the eye, and then for the heart, and now going back to restoring the motor capability. This is really amazing, and I guess like you wonderfully presented that quote. It's not stopping here. It's the possibilities. The more you look into it, the more ideas you get. So wonderful representation. My colleagues are telling that we have reached we have exceeded 130 guests. Thank you very much, guests and colleagues. That's really a testimony about the excellent work that you've done, the interesting topic. So that's really amazing. Of course, I guess, and this is I checked with my colleagues that we will indeed publish the video and obviously the slides. We have quite some questions, so I will try to go over them, and then I will hand it over to you. So we have a guest that is a neuroengineer student and would like to do an internship or a PhD, and is wonder how he can contact you, Professor.
Oh, it's pretty simple. You Google me and you send me an email. silvestro.michera@epfl.ch or at santanapisa.it. And in general, I reply to everybody. So please send me an email, and with pleasure I will reply.
Great. A guest is asking for the reference about the Danish patient test.
Yeah, there are many papers. The first one I would suggest to read is called Raspopovich and others in Science Translational Medicine 2014. But again, if you need more information, just drop me an email and I will send you more papers.
Another guest, Ioannis, is asking for suggestions for materials or papers to read if one would like to deepen a bit this amazing field.
There are few reviews. Again, now it's becoming too many maybe, but drop me an email and I will send you a bit of indication. There are a couple of review papers which could be a very good starting point. We wrote one, other colleagues wrote a couple of them. So and we have now another one just submitted. So drop me an email, and one, for example, we wrote with Grégoire Courtine and Dave Borton and José Millán in 2013. So it's Borton and colleagues in Science Translational Medicine 2013. It would be a good way to start with. But drop me an email and I will ask you. I will send you more information.
Thank you, Professor. We have a question. I think it was put more in the beginning of the presentation, because I think you hinted to that in the second part. Do you use any neural network? No, sorry, that was the previous one. Did you trial with patients with peripheral neurological issues, sensory/motor, but not missing body parts?
We are starting. So the idea, for peripheral, no, but the idea is to have people with spinal cord injury implanted with electrode peripherally for the restoration of grasping movements. We have Loredana, the last patient, as I mentioned, she had limited usability of the medial nerve, and we found, so clearly if you want to implant an electrode in the nerve, if the nerve is damaged, could be complicated. So clearly you need to have a reasonably in good shape nerve to implant.
Thank you. Another question: do you use any neural network or machine learning as an interface between the biological and the electronic parts?
Machine learning is crucial for us for the control. So all the stuff that I showed you for vagus nerve recording, for EMG, for muscular recording, for controlling the prosthesis, they're all based on machine learning, neural network, genetic algorithms, you name. Also for the optic nerve, we just submitted a paper on a deep learning approach for optimizing the stimulation. Machine learning is definitely a crucial part of what we do.
Thank you. Are there any foreseen dangers of this, is asking one of our guests today.
The issue of augmentation of military use or something like related to the Manchurian candidate, in the long term, it is possible. Now, it's not possible for the simple reason that the technology, it's not cool enough. It's a bit primitive, if you want. It works, as you can see, but it is a bit primitive. So Not now, but in the long term, we should keep an eye on the ethics. We should be sure that people are not using this for bad stuff. Not now, but let's say, I would say in 15 or 20 years, this is going to be something to consider and to discuss about.
But do you know if, I don't know, perhaps DARPA is already, because I know they have projects, and they had projects that are a bit similar or in the same field as yours. Do you know if they, I don't know, they are looking into this direction, or they may be even ahead? Who knows?
I mean, there are many projects. The DARPA launched 2 huge projects: the revolutionary prosthetics and the haptics projects. They were huge, and they were providing a lot of results from colleagues in the States. The application in this case was always for disabled people.
Yes.
The trigger was the war in Afghan, the war in Iraq.
Yes.
All the soldiers who came back with
IEDs.
So it could be that in the long term they could also think about something, let's say, really military. But as far as I know, they are really focusing on disabled veterans.
Another question: what are the psychological criteria for the success of these experiments, e.g., acceptance by the patients?
You need to have very motivated patients, especially at the very beginning, for 2 reasons. I mean, this is a technology that they know, and we explain them very well, will not be immediately usable by them in the everyday activities. So they have to be motivated and understand that they are really donated part of their life science for them and the other amputees in 10 or 15 years from now. So this is very important. This is, as I was saying before, many years ago, a colleague of mine gave me a book called Pioneers. And there was not people like me. They were the first patient with a cochlear implant, the first patient with a brain-machine interface. They are really the pioneers. And you have to find people who have, of course, a very good neural condition. As I told you before, they need to have a good peripheral nerve, these kind of things. But they have to be also motivated to work with us. It could be a bit tiring, honestly. We try to be very respectful. So if the patient says, I mean, today I don't want to do it, or this morning I don't want to do it, we don't do it, of course. But even in that case, I mean, Dennis performed more than 700 trials in 1 week. So he was really spending with us, 12 hours in a row, kind of. I mean, this was really too much, but in general, you need to be very motivated.
Professor, another question that shows the interest of the topic. So for the work done on lower extremities, how was the metabolic cost during walking measured?
Oh, there was the standard procedure using recording the CO2 during the movement. It was the standard clinical test they performed. And the interesting thing was that all the parameters, including the metabolic cost, was really significantly reduced during the use. But it was the standard one, nothing really special.
Okay. Another question, a bit of a special one. I have MS, capital M, capital S, more than 20 years. I only need the brain. The rest can be artificial. Are in the field projects for people like me?
Multiple sclerosis is it could be more complicated. And in general, there is not much you can do right now for using our approach. As I said, because multiple sclerosis is damaging exactly, among the other things, what we are doing. So not this kind of technology. Probably there will be more solution from gene therapy and these kind of things.
Thank you, Professor. I think we arrived more or less at the end. Just quite a few reactions, and thank you for your feedback about the topic. Fascinating to hear about these developments. Fascinating overview and details. again, recording, asking for the recording, and how many years do you, another question, how many years do you believe modifying brain activity to treat disorders such as Alzheimer may become reality?
Alzheimer could be complicated, again, because the idea is neural engineering works when you are kind of interfering with the nervous system which is not damaged. In the long term, Alzheimer generates huge damage in the nervous system, which becomes very difficult to interact with this kind of disorder. But there are a lot of evidence, growing evidences that for other cognitive issues, neural engineering could be a very good solution. For example, there is a paper published in Nature Medicine a few weeks ago about neural modulation at the cortical level against depression, with preliminary but interesting results. So clearly, neuromodulation at the cortical level is already used for Parkinson patients to restore a good quality of with deep brain stimulation. And the same idea of implanted electrodes for cortical neuromodulation probably could help in depression or other kind of disorders. But Alzheimer is a bit more complicated, I'm afraid, with neural engineering.
I mean, I remember that we had another FED seminar quite some years ago that was exactly a project with, I think it was coordinated by CNRS, if I'm mistaken, which was targeted towards this disease, and it was really mind blowing. So if there is interest, I'm just reacting of the interest of our guests. If there is interest, and please do not hesitate to mention this in the slide or in the feedback, so that we gather if there is we can present other projects that deal with diseases like Alzheimer. Okay, so please don't hesitate to mention your interest, and then we will schedule this in the future. Till now, we mostly disseminate this information inside, but as today demonstrate, there is a huge interest about the type of projects that we are doing, really close to science fiction for outside audience. So again, if you are interested in the technologies, of course, emerging about that could potentially treat disease like Alzheimer, don't hesitate to leave your feedback, and we'll do our utmost to put this in the future. And then another question also related to multiple sclerosis: is it possible to digitize human memory?
Probably you heard about Elon Musk and his company called Neuralink. He's working, he moved after revolutionizing car industry and space industry, is working on do the same on neural engineering, which is really exciting. And what they're developing is promising, but the technology we have right now for this is very limited. So there are groups trying to restore memory or to reduce, going back to Alzheimer, to reduce the loss of memory by means of neurostimulation. There are interesting preliminary evidences, but put all your memory in digitally somewhere is complicated and not feasible right now. I mean, there are limits in the technology, in the selectivity of the recording you can do with the implants, limit in the space you need to have to put all your memory somewhere, and limit in the way you can take back the memory from the server. The idea is that then you should be able to decode a memory from the neural signals, which is something we don't know how to do yet. In the future, who knows, maybe 20 years from now.
Another question, we have 2 or 3 more questions, so I think we'll fit the time. From Walter, what are the prospects for linking into the cognitive levels rather than sensory motor?
I mean, as I was saying, it's becoming very popular. There are people working mainly on the neuromodulation of the cortex, non-invasively through the scalp or invasively, and they're starting working on depression, on memory issues. So it is very complicated, of course, right? Because, I mean, memory is something extremely complicated, depression can be many different things, but it is becoming quite popular. Let's say it's another emerging application, clearly, of neuromodulation. For example, there are also people working with the results that are not super good, but to deal with obesity using new cortical neuromodulation. The idea is that you can stop craving, and people don't need to, they need to eat more and more using that kind of stimulation. There is also the first clinical trial, as far as I know, was so and so. But I mean, really, as I said, as Tom Mortimer said, told me many years ago, there are really unlimited possibilities. So not for everything, but for many different things, including cognitive, for sure.
Before last question, do you envision applying this technology to enhance our interaction with virtual environments? Very topical question.
It is, and it is, it is very interesting. It's kind of related to the augmentation part. So the idea is, yes, there are many people already working on the idea of making our immersion into the virtual world more realistic and more natural. In general, they do use non-invasive techniques, non-invasive systems,
I hope
for obvious reasons, which limit a bit the performance, but they can already do something immersive, pretty realistic. In the long term, going back to Elon Musk, there is the idea of using implants in humans, healthy humans, to achieve the same kind of things, but then you need to have an implant like Matrix, the movie, something which is extremely easy to implant with no damage and these kind of things. Not for now. In general, there is immersive technology, but definitely non-invasive, which works.
And last but not least, really the last question, what about neuropathies, where the motor nerves are affected? For example, the myelin is damaged. Is there something that can be done using this technology?
If you have pain, yes. For example, phantom limb pain in patients with amputation is pretty common, and you can significantly reduce phantom limb pain by stimulating. Because in amputees, the pain is due to the absence also of sensory feedback. If you restore the sensory feedback, the brain gets something familiar and the pain goes down. The neuropathy, so if there is a problem in the nerve, again, becomes complicated because then you are affecting the gate you are using for the stimulation. So it could work. For example, there are papers showing that you can slow down the degeneration of the nerve by stimulating, because you are generating kind of activity like physical exercise, but with certain limitations.
Professor, thank you very much. We exhausted all the questions. I think we are perfectly in time, 2 minutes, 1 minute to go. So once again, really thank you very much for these exciting insights. I think we had excellent feedback, and I would like to invite you also for the future to see how the results have been progressed, especially with new heart and the new avenues for researchers. I thank you also very much to my colleagues from Connect University for inviting us to Connect University, and I hand the mic over to them. Andrea, thank you very much.
Thanks a lot.
Professor.
Great pleasure.
Many thanks to both of you, and thanks also to the participants for the very engaging questions. We will definitely organize more sessions as such in the future, as we see that there is a big interest. Meanwhile, don't forget to join the next Connect University this Thursday on the 28th, when we will discuss more about your rights when using online services in view of the new set of the rules for the all digital services. So thanks again to all of you, and see you on Thursday.
Thanks a lot. Bye-bye.
Looking forward. Bye.
Many thanks. Bye-bye.